Assalamualaikum warahmatullahi wabarakatuh. Uh, Today, our group would like to present one of our topic, which is Herpedu bumi, or its scientific name is Andrographis Paniculata. So here the group members: Muhammad Aiman Syafiq bin Muhammad Afzanizam, Muhammad Fakhruddin bin Daud Ilyas Ibrahim, which is me, Adib Akmal bin Muhammad Yazid, Arisa binti Muhammad Arshad, and Nur Syafika binti Muhammad Nur. So here the taxonomy hierarchy of Hempedu bumi. Okay, next. Habitat of Andrographis paniculata. Andrographis paniculata herbs, commonly known as kalme, chuan kian lin, or king of bitter, and grim chireta. In Malaysia, it's known as Hempedu bumi. Andrographis paniculata is distributed in tropical Asian country, often in isolate patch. Native population of Andrographis paniculata are spread throughout South India and Sri Lanka, which perhaps represent the center of origin and diversity of the species. Andrographis paniculata is an annual plant, branch, erect, and herbaceous plant which grows in hedgerow throughout plain land, hill slope, waste ground, farm, moist habitat, seashore, and roadside, according to Sanawar Hussain. Also, according to Sanawar Hussain, more shady place, forests, and wasteland are preferable for their well development. The last one is the growing media, which commonly used for growing Andrographis paniculata are soil and alluvial soil. Alluvial soil can be found at the river, which their soil are moisture, while soil they are more like sandy soil. Okay, so the next topic is propagation method of Andrographis paniculata, which will be represented by my friends.
These are the three common propagation methods of Andrographis paniculata. The first one is germination by seeds. Similar to other seed germination, first, seeds are soaked in water for 24 hours and then sown in the soil. After that, transplanting of young seedlings can be done after 6 weeks of germination stage. The next propagation method of Andrographis paniculata is by propagation of stem cutting. In this method, which is propagation by stem cutting, matured stems of mother plant are slant cut within 5 cm to 7 cm long. Then, stems are dipped into rooting hormone to stimulate roots growth. The stems will be transferred into planting bed with 100% sand media, and then plants that survive will be transferred into big pot for smoother growth. The next method is by in vitro propagation, which is tissue culture. In this method, shoot tips and nodal segments from Andrographis paniculata are collected and they are washed thoroughly. Then, shoot tips and nodal segments are then surface sterilized. Shoot tips and nodal segments are divided into small pieces and then are cultured onto culture medium. Lastly, the cultures are regularly subcultured at 3 weeks intervals on fresh medium and then allowed to undergo hardening process.
Next, the secondary metabolite in Andrographis paniculata. The whole plant A paniculata plant is actually therapeutically important, where the leaf contain the maximum amount of terpenoid, including andrographolite, followed by stems and roots, while least found in the seeds. For the concentration and composition of phytochemical in plant are different due to seasons, phenological stage, plant part use, and geography region of cultivation. The presence of lactone, andrographolite, and calmic are actually the main principle for the bitter taste characteristic of A paniculata. Andrographolites are the most abundant compound and responsible for most bioactivities. Flavonoids mainly exist in roots but also been isolated from the leaf of A. paniculata. Aerial part of A. paniculata contain alkanes, ketones and aldehyde. Table 1 shows some major compounds identified in different extracts of various parts of A. paniculata plants. According to Sare et al. 2012, there are more than 80 phytocompounds found in this plant. This table shows some of them. It is grouped by two, which is andrographolite types and non-andrographolite types. Table 2 shows some of the phytocompound and its beneficial activity. For your information, most of the phytocompounds found in this plant are reported to have beneficial effects on human health.
For secondary metabolite analysis of Andrographis paniculata, we use nuclear magnetic resonance stereoscopy, or we call it as NMR stereoscopy. First step of this analysis is leaf samples are quenched in liquid nitrogen and further pulverized into powder form in the cryogenic state. Second step is the sample are freeze drying over 48 hours to remove residual moisture in the leaf tissues. The third step is for each dry sample, directly extract with prepared deuterated methanol as solvent and monopotassium phosphate as buffer in heavy water contain pH 6.0 containing 0.1% 3-trimethylsilyl propionic 2233D4 acid sodium salt TSP and vortex using a vortex mixer. Next, the direct extraction was further assisted by 30 minutes sonication in sonicator bath at ambient temperature. The clear deuterated supernatant obtained after centrifuging for 10 minutes was transferred into NMR tube for measurement of its spectrum. The NMR nuclear magnetic resonance measurement were performed using 500 MHz variant Innova NMR spectrometer operating at about 500 MHz with the temperature is maintained at 26 degrees Celsius. We use nuclear magnetic resonance NMR stereoscopy is to use detect the compound contained in the Andrographis paniculata. Okay, this is a diagram of the NMR stereoscopy.
After that, I will share about the health benefits of Andrographis paniculata. Andrographis is most widely used to treat cold and flu symptoms. Andrographis is also said to act as a natural immune booster. The herb has been used to treat certain other conditions. These include cancer, rheumatoid arthritis, allergies, upper respiratory tract infections, which found to shorten the duration of cold, sore throat, and sick days compared to the standard care. Ulcerative colitis, which used for herbal remedies in the treatment of ulcerative colitis and Crohn's disease. And lastly is multiple sclerosis, which Andrographis may help to reduce fatigue in people with multiple sclerosis according to a small study published in BMC Neurology. That's all from us. Thank you.